Welcome back everyone to MMBS111 Introductory Microbiology. This mini lecture is going to focus on energy. We have 6 learning outcomes for this mini lecture. First, name the energy storing molecule in cells. 2, explain the difference between endergonic and exergonic reactions. 3, define oxidation and reduction. 4, create a general diagram of a redox reaction. 5, name the 2 electron carriers used in cells. And finally, define phosphorylate. So we have some technical terms in this presentation. And hopefully we'll get through them all and you'll have a much better understanding of those terms. So we're talking about energy in cells in this section, and energy in cells is primarily stored as ATP. There are a few other energy molecules, but the majority of energy is stored as ATP. We manage energy within the cells through chemical reactions that involve making bonds and breaking bonds, and the transfer of electrons from one substance to another. So an exergonic reaction is a reaction where energy is released, and that energy then becomes available for cells to do work, to perform their normal functions, okay? And that energy release generally comes in the breaking of a bond, typically the final bond between the terminal phosphates of ATP. Endergonic reactions work in the opposite. These are reactions that are driven by the addition of energy, so they require energy in order to happen. Exergonic and endergonic reactions are typically found together or coupled, so that the release of energy from the exergonic reaction can immediately be put to work in the endergonic reactions. If you look at the diagram there on the right, you can see there are several examples of exergonic and endergonic reactions. Cellular respiration and catabolic reactions are all exergonic reactions. They release energy. Endergonic reactions include things like active transport, cellular movements, and any other anabolic reactions. Oxidation and reduction is another way that energy can be produced, and that's through the movement of electrons. Oxidation involves the loss of electrons. So when a compound loses electrons to another compound, that compound becomes oxidized. Reduction involves gaining electrons. So a compound that gains electrons is said to be reduced. So if you look at the image on the right, you can see we have compound A and compound B. Compound A is said to be the reducing agent. It has extra electrons that it's going to give or lose to compound B, which is said to be the oxidizing agent. So when compound A loses its electrons to compound B, it is said to be oxidized. And when compound B gains those electrons or takes those electrons from compound A, it is said to be reduced. Oxidation reduction reactions are very common in cells, and they are essential for energy transformations. One part of cellular respiration and producing energy involves almost solely oxidation reduction reactions. A redox pair is a pair of compounds that involve a compound that donates electrons and a compound that accepts electrons. So we would say A and B are redox pairs because one donates the electrons, the other accepts those electrons. There are several electron carriers that can be found within the cell. These are oxidoreductases. Okay, these are enzymes that remove electrons from one substrate and add them to another. Some electron carriers that we're going to become very familiar with in this class are NAD and FAD, or nicotinamide adenine dinucleotide and flavin adenine dinucleotide. Okay, we just refer to them as NAD and FAD, or NAD and FAD. It's much easier that way. The energy then that is present in these enzymes as electron acceptors can then be captured to phosphorylate ADP into ATP. So that final bond that adds that additional phosphate to ADP to form ATP, energy is then stored in that terminal bond. So let's look at ATP itself and its role in metabolism and energy. ATP utilization and replenishment is a constant ongoing cycle within cells. Cells are regularly using ATP and forming ATP. When ATP is used, that terminal phosphate is removed from ATP. When that bond is broken, energy is released and we form ADP. When ATP is replenished, that terminal phosphate is restored and the bond is formed between that second and third phosphate. Energy is stored within that bond to be used in later reactions. In heterotrophs, catabolic pathways provide the energy that generates that high energy phosphate to form ATP from ADP. We rarely go beyond that terminal phosphate. Some reactions do use ADP and form AMP, and there is some energy that is stored in that second bond between the first and second phosphate. However, it's not as much energy. So most cellular reactions are only going to use ATP and form ADP. And that is the end of 7.2. So to check in, make sure that you can respond properly to these questions. Go back and review the learning objectives. Our critical thinking question involves glycolysis. The final stage of glycolysis involves the following: phosphoryl pyruvate converted to pyruvate with the addition of ADP to end forming ATP. So is this considered an exergonic or an endergonic reaction? In order to answer this question, you need to understand the difference between an exergonic and an endergonic reaction. So I'd encourage you to go back to those slides and make sure you understand the difference between the two so that you can answer this question. And I hope this video was helpful.